Hello students, this is Dr. Ravi Saini. Our today's discussion is about the neuromuscular junction. So here we are having three terms. Neuro means nerves, muscular means muscle cells, and there is a junction between these two cells. So this junction, so there is a junction between nerves and muscle cells, which is called neuromuscular junction. In previous classes we have studied about uh, that there is a junction also present between nerves and nerves, or neuron and neuron. So we called it synapse. Here we are having a junction between nerves and muscle cells. So basically it is a chemical synapse. It could be called chemical synapse because there is a chemical neurotransmitter which is responsible for uh, coordination between nerves and muscles. So the junction between terminal branches of the nerve fiber and muscle fiber is called neuromuscular junction. Each nerve fiber axon divides into many terminal branches. In this image, this one is the axon of neuron or nerve cells, and it is dividing into axon terminals. And this terminal branches innervates one muscle fiber or muscle cells through this neuromuscular junction. This one is the muscle cell, and these axon terminals are embedded into this muscle cell and forming a connection which is called neuromuscular junction. Now come to the various structures involved in neuromuscular junction. The axon terminals, a synaptic trough or cleft, and the motor end plate. This one is the axon terminal. This one is a motor end plate. So basically, motor end plate it is a modified membrane of muscle cell. There is a space between these uh, two membranes. This is the presynaptic membrane. The membrane of this axon terminal is the presynaptic membrane, and this modified membrane of muscle cell is motor end plate. It is a post-synaptic membrane. There is a space between these two membranes. It is called synaptic trough or cleft. Now the neuromuscular transmission. So there is a transfer of information from motor nerve ending to the muscle fiber through this neuromuscular junction. But this information is in the form of electrical signals, and we know that this electrical signal, it is called action potential. So basically action potential, it is any electrical change across the membrane, and this electrical change is called action potential, and it is got transferred from motor nerve ending to the muscle fiber through this neuromuscular junction. And this uh, neuromuscular transmission involves these events. The first one is the release of acetylcholine, action of acetylcholine, development of end plate potential, generation of action potential in muscle fiber, which is responsible for the muscle contraction. And in the last, there is a destruction of acetylcholine. These are the various events. Now come to the first event, which is release of acetylcholine. So there is an action potential which is traveling from motor nerve fiber, and when this action potential reaches to the axon terminals, it causes opening of voltage gated calcium channels. These are voltage gated calcium channels because this action potential, basically it is an electrical difference or potential. This is responsible for the opening of these voltage gated calcium channels, and when these channels opens, there will be entry of calcium ions from the ECF into the through these channels, and this calcium is responsible for the opening of the synaptic vesicles which were present in the axon terminals. This one was the axon terminal, motor end plate, and there are synaptic vesicles. And when the action potential is reaching towards the axon terminal, there will be opening of calcium channels. Entry of calcium channel is responsible for the opening of these vesicles, and these vesicles contain acetylcholine. This acetylcholine releases in the synaptic cleft and reaches to the motor end plate or the post-synaptic membrane. Finally, there is a passage of acetylcholine in the synaptic cleft. The second stages are the second events are the action of acetylcholine and development of end plate potential. When action potential reaches to the axon terminals, it causes opening of calcium channels, voltage gated calcium channels. So there is a influx of calcium, and this calcium got bonded with the synaptic vesicles, and it is responsible for the release of acetylcholine from these synaptic vesicles to the synaptic cleft, and this acetylcholine finally reaches to the post synaptic membrane and binded with the receptors and forms acetylcholine receptor complex. So there is a motor end plate, axon terminal, and this acetylcholine releases in the synaptic cleft. It got attached with the receptors, and this binding of acetylcholine with these receptors is responsible for the formation of acetylcholine receptor complex, and it causes opening of ligand gated sodium channels, because when the opening or closing of the channel is influenced by the attachment of any chemical compound, we call them ligand gated sodium channels. So in this case, there's an attachment of acetylcholine with the receptors, and these receptors are responsible for the opening of ligand gated sodium channels. When these sodium channels will open, there will be a influx of sodium ions from the ECF. And this influx of sodium ion is responsible for the generation of end plate potential or the motor end plate potential. It is in strength, it is less than the action potential. Basically, a new term arrives here, which is, we are calling it graded potential. As its name suggests, graded. So there is a gradual increase. When the intensity of stimulus is increased, the response of the action potential is also increases. It is called graded potential. This graded potential is responsible for the generation of full flash action potential. It is mild or local change. We are calling it mini end plate potential, because these voltage, these ligand-gated sodium channels are less in number than the voltage-gated sodium channels, and these are not responsible. These are having less sodium influx than the voltage-gated sodium channels. In case of voltage-gated sodium channels, there is a heavy influx of sodium ions. So these ligand-gated sodium channels are responsible for the generation of mild or local potential. It is called graded potential, but in case of neuromuscular junction, we are calling it motor end plate potential. This motor end plate potential will cause generation of action potential in muscle cell. Now there is a generation of action potential in muscle fiber, which finally causes muscle contraction. When acetylcholine binded with the receptor, it causes opening of ligand-gated sodium channels. These channels have less sodium influx and are responsible for the generation of motor end plate potential, which is basically a graded potential. And this graded potential or motor end plate potential finally causes opening of voltage gated sodium channels. And these voltage gated sodium channels are large in number and causes heavy influx of sodium. Now there will be generation of action potential. So finally, there is a transmission of action potential from nerves to the muscle cell. And this action potential causes muscular contraction. In previous classes, we studied about the sliding mechanism. When this action potential reaches to the T tubules, there are T tubules which are formed by the invagination of sarcolemma. This one is a T tubule. Here we are having invagination of sarcolemma. So action potential is traveling from both the sides and finally entering to the T tubules and evenly spread or spreads very and it spreads very fast with these T tubules and reaches to the L tubules. So at the junction of T tubules and L tubules, there are calcium channels, and this action potential is responsible for the opening of these calcium channels, and it causes calcium influx. Finally, this calcium got attached with the troponin protein of thin filament. In muscle fiber, we have thin filament and thick filament. And the thin filament is formed by the three proteins, troponin, tropomyosin, and the actin protein. And this calcium is attached with the troponin protein and pulls away the tropomyosin. And finally, it causes opening or freeing of the active site of the active site of the actin protein. Basically, in case of thin filament, we are having three proteins: troponin, tropomyosin, and the actin protein. This calcium is getting attached with the troponin protein and causes pulling away of the tropomyosin protein. And finally, actin protein have a free active site, and this active site is got attached with the myosin head. When the myosin head got attached with the active site of the actin protein, there is a formation of cross bridge, and this cross bridge causes a stroke by the help of generation of ATP, and this tilting of myosin head pulls actin filament from both the sides towards the center. From both the sides towards the center, there is a myosin head. It is getting attached with this actin protein and is responsible for the pulling this actin filament towards the center. Same thing is happening here. This is the myosin head getting attached with the actin active site of the actin protein and is responsible for the pulling of this actin filament towards the center. So basically, these myosin heads reducing the width of the sarcomere because there are Z lines on both sides, and these Z lines got reduced when the myosin head pulls these actin filaments, and finally there is a contraction of muscle. So finally, the action potential which is generated in the muscle fiber causes muscle contraction by the help of sliding mechanism.